Basics of Medical Biochemistry. First edition, 2022. Chapter 1. Introduction to Cell, Biochemical Point of View. Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Dow, M.Phil Philosophy, Ph.D., Dip M.E. 1.4 Cell membrane. The cell membrane is a very thin, elastic, and delicate envelope present around all animal cells. It is regarded as the barrier between ECF and ICF, an important component of the homeostasis system of the body. Regulator to maintain the composition of ECF and ICF at a normal level. Thickness, 8 to 10 nanometers or 80 to 100 A, 1 nanometer equals 10 to 9 m, 1 A equals 10 to 10 m. Composition, proteins 55%, mainly glycoproteins, lipids 42%, phospholipids 25%, cholesterol 13%, others 4%, and carbohydrates 3%. Lipids of membrane. The lipid is the main component of the cell membrane. As indicated by the lysis of membrane by lipolytic agents such as ether, chloroform, and benzene. The main membrane lipids are phospholipids, cholesterol, and glycolipids. Membrane lipids are amphipathic in nature because they possess both hydrophilic and hydrophobic regions in their molecule. The hydrophilic end, which is the phosphate group in phospholipid and hydroxyl radical in cholesterol, line the external and internal surfaces of the membrane. The hydrophobic end, which is the fatty acid chain in phospholipid and steroid nucleus in cholesterol, faces each other in the middle of the cell membrane. It is 2 molecular thick, so-called bilayer lipid membrane. These lipid bilayers are oriented with their hydrophobic tails inside, while their hydrophilic polar heads are projected externally as well as internally on the surface and are in contact with the aqueous medium on both sides. Forces such as van der Waals, electrostatic, hydrogen bonds, and non-covalent interactions are all forces that contribute to the formation of the lipid bilayer. Overall, hydrophobic interactions are the major driving force in the formation of lipid bilayers. The lipid bilayer is in the form of fluid, which is the basis of a fluid mosaic model of the cell membrane. Figure 1.7 Structure of cell membrane. Source: biologydictionary.net. Proteins of membrane. Membrane proteins are found as floating globular masses within the lipid bilayer. Most membrane proteins are globular, amphipathic, and bound tightly to the membrane. They are the following 2 types. I-integral proteins. These are an essential component of the cell membrane and are also known as intrinsic membrane proteins. These are deeply embedded and traverse the whole thickness of the cell membrane. They are also called transmembrane proteins. They are kept in position by van der Waals forces of attraction within hydrophobic regions of the cell membrane. They possess active sites on both the ends and worked as channels, through which small soluble substances can diffuse. Carriers. These are used to transport the materials across the cell membrane. 2 peripheral proteins. These proteins are present on either the external or internal half of the cell membrane. They are also called surface proteins. These are weakly and reversibly bound to the hydrophilic regions, polar heads, of the membrane lipids as well as with specific integral proteins. They are held in position by ionic interactions or hydrogen bonds. They worked as receptors, when they are present on the outer half of the cell membrane and provide sites of receptors for antibodies, hormones, neurotransmitters, growth factors, and other pharmacological agents. Enzymes. These proteins are present on the inner half of the cell membrane and used to activate or inactivate the various substances. Carbohydrates of membrane. Carbohydrates in the membrane are very less in amount. Usually, they are present as carbohydrate portions of glycoproteins and glycolipids. They are found as protrusions on the surface of the cell membrane. They provide receptors for some hormones, or sometimes they help in various reactions of immunity. Fluid mosaic model of biological membrane. The fluid mosaic model was proposed by S. Jonathan Singer and Garth Nicholson in 1972. It represents the overall organization of biological membranes. The essence of their model is that, the membranes are the two-dimensional solution of oriental lipids and globular proteins. This proposal has been supported by a wide variety of experimental observations. Most of the membrane phospholipids and glycolipids molecules are arranged bilayer. This lipid bilayer has a dual role. It is a solvent for integral membrane proteins and acts as a permeability barrier. Different membrane lipids interact specifically with particular membrane proteins which are essential for their function. One molecule of phospholipid can move several micrometers per second within the plane of the membrane. The proteins are irregularly distributed in the lipid bilayer and give the mosaic appearance to the cell membrane. The membrane proteins are free to diffuse laterally in the lipid matrix unless restricted by special interaction. The floating peripheral membrane proteins look like an iceberg floating in the sea. Effect of temperature and fluidity on the fluid mosaic model of cell membrane. There is a direct relationship between temperature and fluidity of the membrane. As the temperature increases, the fluidity also increases. It is due to the melting of hydrophobic chains of fatty acids. The temperature at which the hydrophobic side chains undergo a transition, melting, from the ordered state, crystalline, to the disordered state, liquid or fluid-like, is called transitional temperature, Tm. The longer and more saturated fatty acid chains interact more strongly with each other and thus required a higher value of Tm. The functions of the cell membrane are significantly affected by its fluidity. As membrane fluidity increases, its permeability to water and other small hydrophilic molecules also increases. The lateral mobility of integral proteins is found to be rapid at high fluidity. Lipid rafts and caveoli. Recently conducted studies showed that the lipid bilayer has two characteristic structures at certain points in the fluid mosaic model called lipid rafts and caveoli. The lipid rafts are composed of clusters of sphingolipids with cholesterol in the outer layer of the plasma membrane. They can float on more fluid phospholipids. They have receptors and signaling proteins and are involved in signal transduction. The caveoli are derived from lipid rafts and are found and formed on inward curving of the inner layer of the plasma membrane. They have a special protein called caveolin 1. They provide rigidity to the cell membrane. Other proteins detected in caveoli belong to the insulin receptor, G proteins, folate receptors and endothelial nitric oxide synthase as a component of their transduction system. The cell membrane of red blood cells. The cell membranes of red blood cells have some special features like specific proteins to assist the special functions performed by erythrocytes. Glycophorin and band 3 protein are the integral proteins of the red cell membrane. Glycophorin is a glycoprotein having 60% carbohydrates, particularly oligosaccharides. The glycophorin provides sites for the attachment of influenza virus while their oligosaccharides component act as antigens of M and N blood groups. The band 3 protein is a dimer of two long and very much folded polypeptide chains. It traverses the whole thickness of the red cell membrane several times. The C and N terminus of this protein is found on the cytosolic side. It worked as a transport protein and provide a channel for the movement of HCO3 and Cl ions. Spectrin, actin, ankyrin, and band 4(1) protein are the important peripheral proteins of the red blood cell membrane. Spectrin is a fibrous protein made up of alpha-beta dimer. It is also linked to actin and band 4.1 protein to form alpha-2 beta-2 tetramers. Actin is found in RBC and other non-muscle cells. Actin filaments are formed by the aggregation of 20 actin molecules. It is involved in the formation of the cytoskeleton of the cell. Ankyrin has two domains, one is attached to the spectrin and the other to the N-terminal of the band 3 protein. In this way, ankyrin provides sites for the attachment of all other peripheral proteins to maintain the cytoskeleton of the red blood cell membrane. Clinical aspects. The normal biconcave shape of RBCs may be altered due to mutation in the gene coding for proteins of the membrane. Two important conditions encountered are hereditary spherocytosis, RBCs become spherical, and hereditary elliptocytosis, RBCs become ellipsoidal. Figure 1.8 Organization of the erythrocyte membrane. These defects result in fragile RBCs with a shorter lifespan. These altered shape RBCs lead to increased hemolysis, anemia, and jaundice. 1.5 Functions of the cell membrane/transport system. The cell is the basic structural and functional unit of all living organisms. To maintain its integrity as a living one, the cell is carrying out certain chemical processes which require different reactants. The products thus formed are either to be stored within the cell or to be transported out of the cell, as excreta. So, there is a need for the transport of various substances into and out of the cell. This necessity of transport is fulfilled by a selectively/differentially permeable membrane called a cell membrane or plasma membrane. The cell membrane is selectively permeable to substances that have the following properties. Lipid-soluble substances. Small-sized particles. Neutral particles. In addition to this category, the cell also needs certain substances to be transported, that is large-sized, lipid-insoluble, and ionic in nature. These types of substances are transported through specific passages called channels, most of which are guarded by specialized gates. The essential role of all the biological membranes, including cell membrane, nuclear membrane, mitochondrial membrane, etc., is to allow the movement of all compounds necessary for the normal function of a cell across the membrane barrier. These compounds include a variety of substances like sugars, amino acids, fatty acids, steroids, cations, anions and many more. These compounds must enter and leave the cell in proper order to maintain the normal functioning of the cell. 1. Ion channels. These are pore-like structures composed of transmembrane proteins. Actually, transmembrane protein subunits cluster together to form a central pore through which ions can pass selectively. They are functionally unique. Most are highly selective for one ion and few are non-selective. This property is based on the charge of the ion, size of the ion and tendency of the ion to attract and hold water around it. They are either passive or active. I-passive channels, leakage channels. They always remain open. These are located all over the neurons and in the excitatory and conductive fibers of the cardiac muscle cells, etc. Cation conductive channels, for Na+, K+, and Ca2+, are negatively charged within the channel and have an average diameter of 5 to 8 nanometers. 2 active channels. They have gates, which open and close in a controlled manner. They allow impermeable ions to cross membranes at rates approaching diffusion limits. Their activities are well regulated by the presence of gates. They are usually highly conserved across species. Figure 1.9 Ion channels of the cell membrane. Source: news-medical.net. Active channels are further classified into 2 types. A. Voltage-gated channels. Gates are controlled by voltage. These channels are open and close in response to changes in membrane potential. They are found in axon hillux, all along unmyelinated axons and at the node of Ranvier in myelinated axons. Their function is the generation and propagation of action potential. Examples are Na+ influx channels and K+ outflux channels in nerves and muscles. B. Ligand/chemically gated channels. A specific molecule binds to a receptor on its respective type of gate and makes it open. These are found in dendrites and cell bodies of neurons. They have a significant role in the generation of synaptic potentials, incoming signals of the neuron. Examples include Cl channels, which are opened by gamma-aminobutyric acid, GABA, and acetylcholine channels opened by acetylcholine. Clinical aspects. Mutations in genes encoding transmembrane proteins can cause specific diseases. Their activities are affected by certain drugs. 2. Ionophores. These are the lipid-soluble compounds that are synthesized by certain microorganisms. These ionophores contain hydrophilic centers that bind specific ions and are surrounded by peripheral hydrophobic regions. They facilitate the transmission of ions across the lipid bilayer membrane either by combining with the ion or by increasing the permeability of the membrane. These function as shuttles for the movement of ions across the membrane. They disrupt transmembrane ion concentration gradients, required for the proper functioning and survival of microorganisms, and thus have antibiotic properties. They act as a defense against competing microbes. There are 2 broad types of ionophores. I-carrier ionophore. They bind to a particular ion, shielding its charge from the surrounding environment, and thus facilitating its crossing through the hydrophobic interior of the lipid membrane. Valinomycin is an example that is produced by a type of streptomyces. It transports K+ from the cytosol into the matrix and H+ from the matrix to the cytosol, thereby decreasing the proton gradient. 2-channel forming ionophore, they introduce a hydrophilic pore into the membrane and facilitate ions to pass through while avoiding contact with the hydrophobic interior of the membrane. For example, gramicidin transports H+, Na+, and K+. Figure 1.10 Structure of carrier ionophore, left, and channel forming ionophore, right. Source, agscientific.com. 3. Aquaporins, water channels. Aquaporins are tetrameric integral membrane proteins that regulate the flow of water across the cell membrane. They belong to a larger family of major intrinsic proteins, MIP, that form pores in the membranes of biological cells. They are also called plumbing systems for cells. Figure 1.11 Structure of aquaporin, water channel. Source, ScienceDirect.com. There are about 13, AP0 to AP12, different types of aquaporins that have been identified. 6 of them are located in the kidney, and a significant number of aquaporins are also found in RBCs. The most studied aquaporins are AP1 to AP4, whose function is purely water reabsorption from the collecting tubules of the kidney. Aquaporins are the channels that transport only water. At very high speeds, the volume of water transported on each side approach 100 times the volume of the cell itself. Clinical aspects: mutations in the gene encoding AP2 protein may cause hereditary nephrogenic diabetes insipidus. Acquired nephrogenic diabetes insipidus can also result from impaired regulation of AP2 due to administration of lithium salts, as a treatment for bipolar disorder, hypokalemia, hypercalcemia, or a chronically high intake of water beyond the normal requirements. A small number of people have been identified with a severe or total deficiency in AP1. They are otherwise generally healthy but exhibit a defect in the ability to concentrate solutes in the urine and to conserve water when deprived of drinking water. De Vick's disease, neuromyelitis optica, has been suggested due to autoimmune reactions against AP4 aquaporin. 4. Gap junctions, nexus. This is a variety of intercellular junctions in which the plasma membranes of adjoining cells are much closed with an intercellular gap of 2 to 3 nanometers that is permeable to colloidal substances. A gap junction is composed of 2 connexins, or hemichannels, that connect the cytoplasm of the 2 cells across the intercellular space. Each connexin is a protein that contains 4 membrane-spanning helices. Gap junctions formed from 2 identical hemichannels are called homotypic, while those with different hemichannels are heterotypic. In turn, hemichannels of uniform connexin composition are called homomeric, while those with differing connexins are heteromeric. Channel composition is thought to influence the function of gap junction. Gap junctions are thought to provide channels of communication between the adjacent cells, and it has been found that their intercellular gap is traversed by a hollow tube-like structure. They mediate and regulate the passage of ions and small molecules up to 1000 to 2000 molecular weight through a narrow hydrophilic pore that connects the cytosol of the adjacent cells. Gap junctions are particularly important in cardiac and smooth muscles. In both of them, the signal to contract is passed efficiently through gap junctions, allowing the heart muscle cells to contract in a syncytium. Few locations have been discovered where there is significant coupling between neurons in the brain. Structures in the brain that have been shown to contain electrically coupled neurons include the vestibular nucleus, the nucleus of the trigeminal nerve, the inferior olivary nucleus, and the ventral tegmental area. There has been some observation of weak neuron to glial cell coupling in the locus ceruleus, and in the cerebellum between Purkinje neurons and Bergman glial cells. It now seems that astrocytes are strongly coupled by gap junctions. Experimental data showed strong gap junction expression in astrocytes. 1.6 Transport across the cell membrane. The transport or translocation of various substances such as ions, nutrients, waste matters, and drug molecules across the cell membrane can be classified into the following groups. 1. Passive transport, simple diffusion and facilitated diffusion. 2. Active transport, primary active transport and secondary active transport. 3. Osmosis. 4. Endocytosis, phagocytosis and pinocytosis. 1. Passive transport. Passive transport means the movement of biochemical particles, atomic or molecular substances, across the cell membrane without using energy. Particles move randomly and spread out from an area of high concentration to an area of low concentration. Simple diffusion. It is the movement of particles from the region of high concentration towards the region of low concentration. There is no utilization of external energy, i.e., ADP. It is also called the downhill process. The moving molecules utilize their own kinetic energy, as they possess by virtue of their motion. The transport of molecules may also take place in response to the electrical gradient. The smallest molecules move more rapidly than large molecules. It is a continuous process, due to the rapid movement of molecules. Molecules move in an unpredictable direction. Diffusional equilibrium is achieved when net movement ceases till the concentration of solution becomes equal throughout. 2. Facilitated diffusion. Facilitated diffusion is the movement of molecules across the cell membrane via special transport proteins that are embedded within the cellular membrane. Many large molecules, such as glucose, are insoluble in lipids and too large to pass through the membrane pores. Therefore, it will bind with its specific carrier protein, and the complex will then be bonded to a receptor site and moved through the cellular membrane. Facilitated diffusion is a passive process, and the solutes still move down the concentration gradient. 2. Active transport. The movement of molecules, ions, or other particles across the cell membrane against concentration gradient or pressure gradient, or electrical gradient with the utilization of energy is called the active transport process. Particle movement takes place from the region of low concentration towards the region of high concentration across the barrier membrane. It is also called the uphill process. Energy and carrier protein is the essential prerequisite of this process. Active transport of cell membrane depends upon the metabolic activity of the cell. Primary active transport. The active transport process which uses energy from the direct breakdown of ADP, adenosine triphosphate, is called primary active transport or direct energy utilizing transport. The most common example of active transport is sodium-potassium, Na plus K plus, pump or Na plus K plus ATPase. Figure 1.12 Primary active transport, sodium-potassium pump. This process is responsible for the maintenance of a high concentration of K plus and low concentration of Na plus in intracellular fluid. The operation of this pump can be described in the following 3 basic steps. 3. Na plus binds to the carrier protein on the inside of the cell. 2. 2K plus ions bind to the carrier protein on the outside of the cell membrane. 3. Now carrier uses energy obtained from ADP to move 2K plus ions into the cell and 3 Na plus ions out of the cell. Other examples of the active transport process are Ca2 plus, Cl and H plus pumps. 2. Secondary active transport. It is carrier-mediated active transport that uses the energy stored in the Na plus concentration gradient is called secondary active transport or indirect energy utilizing active transport process. It occurs only when extracellular Na plus concentration is higher than intracellular Na plus concentration. Energy is required to establish the Na plus concentration gradient. Examples are the transport of glucose and amino acids. Transport of glucose and amino acid against their concentration gradient takes place by a secondary active transport process. The main steps of this process are: Na plus binds to the carrier on the outer side, where it is high in concentration, then carrier affinity for glucose increases, as a result glucose bind with it. 2. Now carrier undergoes some conformational changes during which Na plus and glucose are exposed to the inner side. 3. As the intracellular concentration of Na plus is low, that's why it dissociates from the carrier. As a result, the affinity of a carrier for glucose also diminishes and it moves away from the carrier into the cytoplasm. 4. But the movement of Na+ out of the cell is an energy-utilizing process. Based on the direction and number of particles involved in the process of moving across the cell membrane, the transport system may be uniport, symport and antiport. Figure 1.13 The uniport, symport, and antiport transport system. Uniport system, it is the translocation of solute particles across a membrane that is not coupled to the translocation of another solute, in either the same or the opposite direction. Facilitated diffusion is also included in it. One example is the movement of Ca2+ from the cytosol into the extracellular fluid. Generally, it is mediated by a carrier protein known as a uniporter, example the glucose transport protein of mammalian erythrocytes. Symport system, it is the movement of two different kinds of particles across the cell membrane in the same direction. Sodium-glucose cotransport from the lumen of proximal convoluted tubules into the epithelial cells lining the tubule is a well-known example. Antiport system, it is also called the countertransport process. It involves the translocation of two solute particles of different natures across the membrane barrier in opposite directions. The classical example of an antiporter system is a sodium-potassium pump, Na plus K plus At posse. Other examples include Na plus H plus antiporter working in kidneys and ClHCO, 3 antiporters operating in RBCs. 3. Osmosis. The process of osmosis is brought about by simple diffusion, but only the solvent is allowed to move from higher solvent concentration to lower solvent concentration, or from lower solute concentration, dilute solution, to higher solute concentration, concentrated solution. Osmotic pressure, the force under which solvent moves from a solution of low solute concentration to a solution of higher solute concentration through a semi-permeable membrane, is called osmotic/oncotic pressure, or the pressure required to prevent the process of osmosis is called osmotic pressure. Units of osmotic pressure, equals CRT. The osmotic pressure of a molar solution of non-electrolyte at 0 C equals CRT, where C is molar concentration, R is universal gas constant and T is absolute temperature. Equals 1 times 0.0821 times 273. Equals 22.4 atmosphere. Or equals 22.4 times 760 equals 17,024 mmHg. The SI unit of osmotic pressure is osmol. 1 osmol equals 22.4 atmosphere. Oregon 17024 mmHg. 1 milliosmol equals 17 mmHg. The osmotic pressure of blood plasma, gastric juice, cerebrospinal fluid, CSF, pancreatic juice, and bile is 300 milliosmol. Biochemical significance of osmosis and osmotic pressure include: involved in the exchange of substances at the capillary bed and lymphatic plexus, regulation of urine formation, reabsorption of CSF, absorption of nutrients in the intestine, the continuous osmotic exchange between RBC and plasma. Extensively used technique in the laboratory and various investigations and research projects. For endocytosis and exocytosis, endocytosis is the process of engulfment of particulate matter or extracellular fluid with its contents by the cell membrane. It is of two types, phagocytosis and pinocytosis. Phagocytosis, phago means feeding and cyto means cell, eating by cell. It is the process of ingestion of large particulate matter by the cell, such as a bacterium, other cells, and tissue debris. For example, neutrophils can engulf 5 to 20 bacteria and macrophages can capture 100 bacteria at a time. This process involves the following steps: formation of pseudopodia, encircling of matter, invagination of the closed chamber, formation of phagocytic vesicles. The phagocytic vesicles fuse with primary lysosomes to form secondary lysosomes. The hydrolytic enzymes of secondary lysosomes digest the contents of phagocytic vesicle into amino acids, sugars and nucleotides, which are then released into the cytoplasm for use in different metabolic processes according to the need of the cell. Factors that favor the phagocytosis include: I. Rough surface. The rough surface of the particle increases the chance of its phagocytosis. 2. Electropositive surface. Normal body surfaces are electronegative, so they repel one another. But most of the dead tissues and foreign particles are positively charged, so they can easily be phagocytized. 3. Identification. The immune system helps in the recognition of the particle to be phagocytized. The antibodies, opsonin, combine with the complement system or are attached to foreign particles and made the process of phagocytosis easy. Pinocytosis. It is the process of ingestion of minute quantities of extracellular fluid and dissolved substances in the form of minute vesicles, drinking by cell. Molecules to be ingested come in contact with the cell membrane by adsorption. Changes occurred in the cell membrane followed by invagination of the adsorbed molecules. The invaginated portion of the cell membrane then breaks away from the surface of the cell and results in the formation of the pinocytic vesicle. The two important types of pinocytosis are fluid phase and receptor-mediated. I. Fluid phase pinocytosis. It is a non-selective random process of uptake of solute from the surrounding extracellular fluid. It is an active process and proportionate to the concentration of solute particles. Figure 1.14. The process of endocytosis and exocytosis. The membrane invaginates internally to form a vesicle containing captured particles along with a small amount of extracellular fluid. The portion of the cell membrane that gives rise to vesicle regenerates to maintain the overall integrity of the cell membrane. The contents of the vesicle are hydrolyzed by the lysosomal enzymes into micromolecules. Some of the captured material and their hydrolytic products may then leave the cell in the form of secretory vesicles called secretory endosomes. 2. Receptor-mediated pinocytosis. It is also called absorptive pinocytosis. It is a selective type of endocytosis as the process starts with the binding of the substances to be ingested with their respective receptors. These receptors are present on the external surface of the cell membrane in the form of pits that are lined by special proteins such as clathrin and adaptin. These protein-coated pits form small coated endocytic vesicles, after pinching off from the cell membrane. The formation of vesicles and internalization need many factors like adapter proteins, phosphatidylinositol and other protein dynamin that help in hydrolysis and release of energy from GTP. These vesicles become early endosomes after losing their clathrin coat. The receptors, clathrin, and membrane fragments are recycled back to the plasma membrane by exocytosis. The early endosomes become late endosomes after passing through the stage of multivascular bodies. The contents of endosomes are now transported to lysosomes, Golgi complex and rough endoplasmic reticulum for final metabolism. Low density lipoproteins, LDL, are also taken up by the process of receptor-mediated pinocytosis. Exocytosis. In exocytosis, transport vesicles migrate to the membrane, fuse with it, and release their contents. The movement of the vesicle is carried out by the contractile elements of the microtubular system of cytoplasm. Exocytosis is also called reverse pinocytosis. Many secretory cells use the process of exocytosis to export their products. Following are the type and fate of macromolecules released by the cells via the process of exocytosis. Some protein molecules get attached to the membrane exterior to become peripheral proteins and act as antigens or receptors for hormones. 2. Some molecules like collagen and glycosaminoglycans are incorporated into the extracellular matrix. 3. Hormones like a parathyroid hormone, PTH, insulin, and catecholamines are secreted as granules packed in respective vesicles. 4. Acetylcholine and other neurotransmitters are also released from various presynaptic neurons. The presence of ADP, contractile elements, and Ca2+ is necessary for both endocytosis as well as exocytosis. Summary. The cell is the basic structural and functional unit of life. There are about 100 trillion cells in the human body. Prokaryotic cell lacks a membrane-bound nucleus. Eukaryotic cells are well-established cells with a complete nucleus. All functions of the cell are controlled by the nucleus, including the process of DNA replication and RNA transcription. Mitochondria are responsible for energy production in cells. The Golgi apparatus is composed of tubules and vesicles. It is engaged in both the processing of glycoproteins and the synthesis of complex polysaccharides. The endoplasmic reticulum is an intracellular membrane system found in all eukaryotic cells. It creates a network of communicating tubules mainly between the nucleus and cytoplasm. The rough endoplasmic reticulum has a large number of ribosomes attached to its surface. A lysosome is a membrane-bound organelle in the cytoplasm of most cells containing various hydrolytic enzymes that function in intracellular digestion. Peroxisomes are small vesicles found scattered in the cytoplasm around the nucleus. They contain more than 50 characterized enzymes and perform many biochemical functions, including detoxification. The cytoplasm is found between the nucleus and cell membrane. Its main function is to maintain the shape and consistency of the cell and to hold the organelles. The cell membrane is a very thin, elastic, and delicate envelope present around all animal cells. Its main component is a lipid, which is 2 molecular thick so-called bilayer lipid membrane. Integral proteins of the cell membrane, also known as intrinsic membrane proteins, worked as channel or carrier proteins. Peripheral proteins of the cell membrane worked as receptors or enzymes. Carbohydrates of the cell membrane are found as protrusions on the surface. They provide receptors for some hormones, or sometimes they help in various reactions of immunity. The cell membrane is selectively permeable to substances that are neutral, small-sized and lipid-soluble. Aquaporins are tetrameric integral membrane proteins that regulate the flow of water across the cell membrane. Passive transport is the movement of biochemical particles across the cell membrane without using energy. The direction of passive transport is from higher to lower concentration. Facilitated diffusion is the movement of molecules across the cell membrane via special transport proteins that are embedded within the cellular membrane. Active transport is the movement of molecules, ions, or other particles across the cell membrane against concentration gradient or pressure gradient, or electrical gradient with the utilization of energy and carrier protein. The process of osmosis is brought about by simple diffusion, but only the solvent is allowed to move from higher solvent concentration to lower solvent concentration. The osmotic pressure of blood plasma, gastric juice, cerebrospinal fluid, CSF, pancreatic juice, and bile is 300 milliosmoles. Endocytosis is the process of engulfment of particulate matter or extracellular fluid with its contents by the cell membrane. References. Exercise questions. MCQ 4. Which of the following cell organelles is responsible to control programmed cell death? Golgi apparatus, lysosome, mitochondria, ribosome, peroxisome. MCQ 5. A 29-year-old woman presented with complaints of blurred vision, weakness, and numbness. She also felt irregular bladder and bowel movements and stiffness of muscles. Relevant investigations confirmed the diagnosis of Devick's disease, neuromyelitis optica, due to autoimmune reactions against certain aquaporin. Which of the following aquaporins is affected in this patient? AP0 aquaporin, AP1 aquaporin, AP2 aquaporin, AP3 aquaporin, AP4 aquaporin. MCQ 6. Which of the following cell organelles is responsible to carry out beta oxidation of fatty acids greater than 18 carbons in length? Golgi apparatus, lysosome, mitochondria, ribosome, peroxisome. Please write answers in the comments. End of Part 2 of 2 of Chapter 1. Thank you.